Hello and welcome everyone to my YouTube channel. Today I'll discuss immunoglobulin M with you. Let's proceed. Immunoglobulin M constitutes of 5 to 8% of serum immunoglobulins. Its normal level is zero point 0.5 to 2 milligrams per ml. Its half-life is about 5 days, which means it is a short lifespan. It rises quickly and declines quickly in the circulation. Sedimentation coefficient of immunoglobulin M is 19S, which means it's a heavy molecule. Its molecular weight is 90,000 to 1 lakh daltons, and therefore it is known as the Millionaire Molecule. So if you're asked which immunoglobulin is known as the Millionaire Molecule, you know what your answer should be. Let's take a look at the structure of immunoglobulin. You can already see the picture over here. So it is a pentameric molecule, as you can see. It's the pentameric immunoglobulin, which is made up of 5 monomeric units. Because it is made up of 5 monomeric units, it has a valency of 10, which means each unit binds to 2 antigen, and therefore 5 units will be able to bind to 10 antigens. If the antigens are too large, then the valency may reduce. Each unit is made up of 2 heavy mu chains and 2 light chains. The light chain can either be of lambda or kappa type. These monomeric units are bound together by disulfide bonds, which you can see here, and a J chain. This J chain is a glycoprotein and it is present in two immunoglobulins, that is immunoglobulin M and immunoglobulin A. So if you are asked which are the immunoglobulins that have a J chain, your answer is immunoglobulin M and immunoglobulin A. The functions of immunoglobulin M. Monomeric immunoglobulin M is a major antibody receptor on the surface of B lymphocytes. It provides protection against microorganisms and large antigens with repeating antigenic determinants on their surface. It can carry out bacterial agglutination, which is 20 times more effective. It does opsonization, which is 500 to 1000 times more effective than immunoglobulin G. Please refer to my lecture on immunoglobulin G for understanding opsonization. It also carries out complement fixation, in which antibodies activate the complement pathway and leads to complement mediated cell lysis of the pathogens. The neutralization of toxins is also carried out by immunoglobulin M, where the endotoxins secreted by bacteria such as tetanus toxin are rendered inactive. This results in neutralization of their toxic effects. Immunoglobulin N deficiency is also associated with septicemia. So if anybody is deficient of immunoglobulin M, it may lead to septicemia. Early antibody. Immunoglobulin M is also known as the early antibody. Why so? Let's understand that. So this is an immune response curve. This is the first exposure of the person with the antigen, and as soon as the body is exposed to this antigen, the primary immune response begins, and after some time immunoglobulin M is released. So the first immunoglobulin to be released when the person is infected is immunoglobulin M, and therefore it's the earliest antibody to be synthesized by the fetus beginning by 20 weeks of age. It's the first antibody produced in primary immune response, and it is seen in early phase of infection. Due to all these reasons, immunoglobulin M is known as the early antibody. Immunoglobulin M are also known as natural antibodies. Now let's understand this. Natural antibodies are produced without any known antigenic stimulus by B cells, and hence they are called natural antibodies. So you know that our body starts making antibodies after stimulus from a pathogen which have antigens. However, immunoglobulin M is one such antibodies which are produced without any antigenic stimulus by the B cells. It acts as first line of defense. It can recognize wide range of microbial toxins, so they protect against a variety of new pathogens. Immunoglobulin M and blood groups. So these immunoglobulin M have also an association with blood group. Let's see what that is. It's a predominant immunoglobulin class for blood groups or the blood group A and B. Small quantities of immunoglobulin A and immunoglobulin G are also present. Now this is a table that is showing blood group A and B. The red blood cells in blood group A has surface antigens of one kind, and such a person will have antibodies in the plasma, and these antibodies are known as anti-B, which are antibodies against blood group B. So this is blood group B, which has another set of antigens on their surface, and their plasma will have anti-A antibodies, which is an antibody against blood group A. So this is the association of immunoglobulin M and the blood groups. Immunoglobulin M cannot cross the placental barrier, and this proves to be helpful. Let's see how. Immunoglobulin M is actually the largest and the heaviest of all the immunoglobulin isotypes found in vertebrates. Because it's pentameric, it cannot cross the placenta, and therefore the fetus is protected from natural antibodies of the mother. The presence of immunoglobulin M in the fetus or newborn indicates intrauterine infection. This is the fetus, and it is connected with the placenta, and these immunoglobulins are unable to cross the placental barrier due to their large size, and this keeps the fetus protected from the natural antibodies of the mother. These are my references for the lecture. Thank you for watching. Like, share and subscribe to my channel. Please comment. Don't forget to hit the bell icon. Thank you once again.